Welcome to Module 3, Expected Outcomes for an Upper Extremity FES System. By completion of this learning module, participants will understand the range of functional outcomes for a neuroprosthesis compared to best available treatments, and understand the evolution in FES technology and reconstructive procedures over time. We're going to start by taking a look at the NeuroControl freehand system. This system consisted of an implantable receiver with 8 epimesial or intramuscular electrodes implanted in the muscles of the hand and forearm. Communication and power to the receiver was accomplished through an inductive transmitting coil placed externally over the implant. The other external components included a control unit and a shoulder transducer that served to translate shoulder movements into control signals used by the system. Typical electrode placement of the stimulating channels included the muscles shown here. The system could be programmed to provide 2 grasp patterns, lateral pinch and palmar prehension. Control of grasp opening and closing was accomplished by graded elevation of the contralateral shoulder. The freehand study cohort consisted of 61 participants enrolled at 15 sites. They ranged in age from 15 to 57 years old, 79% were male, 69% were classified as C5 motor level spinal cord injury, and 26% were classified as C6. The time post-injury ranged from about 1 to 32 years, with a median of 4.5 years. In the outcome measures that follow, evaluations were performed with the freehand on in comparison to off, but it's important to note that at the time of this study, it was typical for freehand cohort to receive tendon transfers at the same time as the implant, so the comparison to off represents the outcome of these reconstructive procedures. Common procedures at that time included FDP synchronization, FDS synchronization, EDC synchronization, the FDS Zancoli lasso procedure, and thumb IP joint arthrodesis. These procedures were meant to ensure that all of the fingers closed together or opened together. The thumb arthrodesis was performed to maximize transmission of thumb force by positioning the thumb IP joint in neutral. So with that background, let's take a look at some of the freehand outcomes, starting with pinch force. The freehand cohort had pinch force measurements taken at study entry and at each evaluation point after surgery, with and without the neuroprosthesis. Pinch force measurements for both lateral and palmar grasps were significantly stronger with the neuroprosthesis on compared to off, and the trend over time showed that these gains were maintained. Note that the error bars here indicate plus and minus 1 standard deviation from the mean. Looking now at the grasp release test, here we see the results of the GRT pre-test, showing the number of objects that could be manipulated pre-surgery, and post-surgery with the system off and on. Prior to surgery, participants could move a median of 1 object on the GRT pre-test, and after surgery at the 3-month visit, participants could move a median of 2 objects with the system off. This gives an idea of the important functional gains that can be achieved with tendon transfers and exercise. An improvement of 1 additional object on the GRT pre-test is considered a clinically meaningful difference. But as you can also see on the far right, with the neuroprosthesis on, participants showed an even greater improvement in function. The median number of objects moved was 6. Looking at the GRT pre-test over time, you can see that outcomes remain consistent over the course of the participant's first year post-implant. In the GRAND clinical trial, the data collection schedule will be similar to this, with the primary outcome measure obtained at the 3-month visit and additional data collection performed at 6 and 12 months post-implant. In the freehand study, participants underwent an assessment of activities of daily living using 2 different methods. There were slight differences in the purpose, methods, and number of participants evaluated. In the ADL abilities test, participants were trained and tested to their highest level of ability in selected tasks, with and without the neuroprosthesis. Each participant was evaluated to determine whether the neuroprosthesis decreased the amount of assistance required to perform each activity. In addition, participants were asked to express their preference for using the neuroprosthesis or not for each task. 28 participants were evaluated using this test. Across all participants and tasks, 50% showed an improvement in independent score, and 78% preferred to use the neuroprosthesis to perform the activities. In the ADL assessment, participants and clinicians identified goal activities prior to implantation. Prior to surgery, these activities were demonstrated according to the methods typically used. At rehab, participants were trained in these goal activities with the neuroprosthesis and then evaluated according to whether they met their goals, whether the neuroprosthesis decreased assistance, and whether they preferred to perform the task with the neuroprosthesis. 11 participants were evaluated with this test. Across all participants and tasks, 81% showed an improvement in performance with the neuroprosthesis. The neuroprosthesis was preferred for 85% of tasks, and participants scored that their goals had been met for 89% of the tasks. The GRAND clinical trial will also look at activities of daily living, using the Canadian Occupational Performance Measure to identify functional goals, and following the training and scoring method of the ADL Abilities Test to characterize improvements in independence and the user's preference for the neuroprosthesis. You'll learn more about that in Module 8. Finally, the freehand study also looked at user satisfaction with the system, as well as patterns of usage over time. A survey was sent out to 34 study participants who were at least 1 year post-surgery and had been using the system at home for at least 6 months. 87% reported being very satisfied with the neuroprosthesis. 88% reported that the neuroprosthesis had a positive impact on their lives. 87% reported an improvement in their activities of daily living. 81% reported improved independence. In terms of usage, participants reported using the neuroprosthesis a median of 5.5 days per week. 15 participants used it 7 days a week, and 5 participants reported not using the device. In the GRAND clinical trial, a similar user satisfaction survey will be conducted at the 6-month visit. Usage data will be automatically captured by the implant and analyzed to characterize frequency of use by participants. Now that you have a sense of what the freehand outcomes were like, let's take a look at the IST system. This was a second-generation FES system developed in the mid-1990s to advance the capabilities of an upper extremity neuroprosthesis. The implanted stimulator telemeter, or IST system, was designed to accommodate up to 12 stimulating electrodes and included 2 channels for myoelectric recording. It continued to feature external components, notably the transmitting-receiving coil, which was placed on the skin over the implanted telemeter and connected to a small external control unit. The additional stimulating electrodes allowed more versatility in creating grasp patterns, as well as addressing the specific functional needs of the study participants. Placement of additional electrodes varied across study participants, but included finger intrinsic muscles to provide improved finger IP extension and MCP flexion, triceps for elbow extension, palmaris longus for wrist flexion, pronator quadratus for forearm pronation, and muscles to provide shoulder stabilization for higher level injuries. In addition, the extra channels allowed duplicate electrodes to be placed in the critical muscles for finger flexion and extension. With these additional electrodes, new grasp patterns could be developed, including the sandwich grasp, which is a light palmar pinch with finger IP joints extended. Pointer is a pattern that extends the index finger while the other fingers remain flexed. And the cashew or popcorn grasp, which is a fine index finger to thumb tip pinch. The use of myoelectric signals from voluntary muscles as a control system for the neuroprosthesis was an important advancement of the IST-12 system that enabled smoother and more natural control of grasp opening and closing. This was achieved by the placement of an MES electrode on either the voluntary ECRL or through tendon transfer of the brachioradialis to the ECRB. A second recording electrode was placed on either the platysma or trapezius muscle to provide a logic signal that could be used to switch between grasp patterns or perform other control functions. In this video, a variation on these control methods is shown, where the participant locks and unlocks her grasp pattern by contracting her ECRL. A total of 12 individuals implanted with the IST-12 system were evaluated. In total, 15 arms and 12 individuals with motor level C5 or C6 tetraplegia were enrolled. Some people received more than one implant in order to provide bilateral function, or in one case to provide additional stabilization of the upper arm and shoulder. The study involved 12 individuals, 9 of which had a single implanted IST-12 system, and 3 had 2 implanted IST-12 systems. 10 out of the 12 participants were male, with ages ranging from 26 to 56 years old, with a median age of 37 years old. The motor level of injury for each arm was motor level C5 in 40% of the cases and motor level C6 in 60% of the cases. The time post-injury ranged from 1 to 21 years, with a median of 5.5 years. As with the freehand study, the IST-12 participants underwent reconstructive procedures. In this cohort, some procedures were done in advance of the neuroprosthesis implant, and others were done at the same time. With the new IST-12 capabilities, the types of reconstructive procedures changed somewhat, with greater emphasis on providing elbow extension and providing thumb flexion by transferring the voluntary brachioradialis. The FPL split was used as a way to provide joint stability during FES while still allowing IP joint flexibility during passive use. You'll recall that the freehand study used multiple synchronization procedures of the extrinsic finger muscles. But by the time of the IST-12 study, that strategy was felt to be unnecessary, as grasps were quite functional without them. Looking first at grasp strength, in these charts, the ranges for the IST-12 system are compared directly to previous data from freehand for both lateral pinch on the left and palmar grasp on the right. The data are shown pre-implant, post-implant with the neuroprosthesis off, and post-implant with the neuroprosthesis on. The freehand data are shown in the lighter colored blocks. The results indicate the same trends of significantly increased strength with FES on for both grasp patterns. The IST-12 cohort showed a higher grasp strength at the outset and post-surgery compared to the freehand. Here we see the results of the GRT pretest showing the number of objects that could be manipulated pre-surgery and post-surgery with the system off and on. Prior to surgery, participants could move a median of 2 objects on the GRT pretest, and that remained about the same after surgery at the 3-month visit with the system off. With the system on, participants were able to manipulate a median of 6 objects, which is nearly identical outcome to the freehand cohort. As with the freehand study, the IST study used the ADL abilities test and usage statistics to characterize functional outcomes and acceptance. While these data are currently unpublished, preliminary analysis suggests similar outcomes to the freehand cohort. For example, with regard to activities of daily living, there were 102 tasks tested across 10 participants, or 13 arms. Certain activities, such as eating with a fork, showed an improvement for 100% of the participants. Out of all the tasks tested, 75% showed improved performance with neuroprosthesis on compared to off. Users demonstrated acceptance of and a strong preference for the IST system, incorporating it into activities of daily living in their home environment. In summary, over the last several decades, upper extremity neuroprostheses have evolved to include more stimulating channels, more grasp pattern options, fewer external components, and improved control of grasp with myoelectric signal processing. You'll see that these trends continue as we introduce the network neuroprosthesis coming up in the next Module 4. As you've also seen, reconstructive procedures have also evolved to better complement the functionality of the neuroprosthesis. Despite these many changes, past studies have been fairly similar in design with respect to the specific choice of outcome measures. And in all cases, functional outcomes have been excellent, with improved grasp strength, GRT performance, activities of daily living, and user acceptance. As we introduce the grand clinical trial in Modules 5 through 8, much of the protocol will feel familiar, as our intent is to build upon the successes of these past studies. You've now completed Module 3. Coming up next in Module 4, we will introduce the Case Western Reserve University Network Neuroprosthesis.